And today we are going to see a most important topic, vacutainer. So what are vacutainers? So vacutainers are used to collect blood. So what are vacutainers? Vacutainers are used to collect blood. So why do you have different colored caps in different vacutainers? That's because each vacutainer has got a different anticoagulant which prevents the clotting of blood. So you can't use the same tube for all the tests like you have for clinical pathology, biochemistry. So you can't use all the tubes for all the tests. So you need certain anticoagulants for certain purpose. So first is a purple or the lavender. Okay, so purple or lavender is one of the most commonly used tube. This contains EDTA, ethylene diamine tetraacetic acid, which is used for estimation of CBC. Most commonly used tube is this, and the most commonly done test is CBC, the complete blood count. Okay, you have to remember the anticoagulant and the purpose. And one more purpose is when you're going to estimate your ESR by Wintrob's method, you can use this tube which contains EDTA. Next is this heparin tube, which is green in color. Okay, so heparin, green color. So heparin, you use it for two conditions. You remember two F's. One is flow cytometry and another one is osmotic fragility. Okay, so flow cytometry and osmotic fragility. So remember, heparin is not used for two conditions, that is two P's. One when you want to see a peripheral smear and another one for PCR. You should not collect in heparin. Why? Because when you want to use heparin for a PS, peripheral smear, it gives a blue discoloration. So it will be very difficult to identify the cells. Likewise, you should not collect blood for PCR purpose in heparin tube because heparin has got certain interaction with restriction enzymes which we use for PCR. So as far as heparin is concerned, it's used for two F's, not used for two P's, used for flow cytometry and osmotic fragility, not used for PCR and peripheral smear. Next is this sky blue colored tube which contains trisodium citrate, usually of concentration 3.2%. Okay, so remember C for C, trisodium citrate for coagulation purpose, and 3.2%, and it's the ratio of 1:9, 1 volume of anticoagulant to 9 volume of blood. So another tube here has got trisodium citrate, which is black colored cap. So this also contains trisodium citrate, but this is of different concentration and different ratio. So this is 3.8% sodium citrate and concentration is 1:4. Okay, so 1 volume of anticoagulant to 4 volume of blood. So why do you need this different thing? Because this is used for coagulation purpose. This black tube specifically is used for ESR by Westergen's method. So ESR by Wintrob's is EDTA, ESR by Westergen's is black colored tube. Okay, so having talked about the black colored tube, I want you to know about the tube with white cap. So I told heparin cannot be used for PCR. So ideally you can collect for PCR in a vacutainer which has got a white cap because that is coated with a polyester. Okay, so black tube is for ESR, white tube is for PCR. Next you have one with a red cap. This contains clot activator. This doesn't contain any anticoagulant. So when you want to separate a serum, you can use it in red tube. So this can separate the blood and the serum or plasma. Okay, so this doesn't contain any anticoagulant. It contains a clot activator. So these are the commonly used tubes. So EDTA for CBC, heparin for flow cytometry and osmotic fragility, sodium citrate blue tube for coagulation purpose, and this gray tube is for estimation of blood sugar levels or blood glucose levels. So what does they contain? They contain a specific thing called fluoride, because when you're going to collect the blood for glucose, your glucose should remain as such. So fluoride is going to inhibit your enolase enzyme which is involved in glycolysis. So once this enzyme is inhibited, your glycolysis will not happen and your blood glucose remains the same. So gray top for glucose, G for G, black for ESR, and red contains clot activator. Thank you.